Okay, so welcome to this video. In this video, what we're going to talk about is circadian clocks. Okay, right, so let me give you an overview of what we're going to discuss in this video. Okay, so we will start off with a basic definition of what a circadian clock actually is. So we'll have a basic discussion of what a circadian rhythm is, what circadian actually means, and what a circadian clock is. Then we'll go on to an example of a circadian clock. Okay, and we'll be talking about the example of a circadian clock present within Drosophila Melanogaster, okay, which is a nice simple example to get us started with. Then what we will move on to is the circadian system within mammals, so within mice and humans, for instance. Okay, and there it's more complicated, as we'll see, okay, because you have this master clock system, which is the suprachiasmatic nucleus, which is keeping all peripheral cells' circadian clock systems in synchrony with the light-dark cycle. Okay, and we'll have a look at what is understood about how that works. Then what I will say is, after presenting you this lovely theory of how the master clock and peripheral clocks all are kept in synchrony with the light-dark cycle, we'll then see that this theory can't be complete, basically, because it all works on these transcriptional translational feedback loops, and I'll tell you exactly what that means. Okay. However, erythrocytes, red blood cells, they have circadian oscillators, circadian clocks. Okay, and yet they don't have a nucleus, so they can't have transcriptional translational feedback loops. Okay, so we'll then talk about how they must have some sort of post-translational oscillator, and to illustrate this point, we will talk about a very well understood example of a post-translational oscillator, which is the cyanobacterial circadian clock system. Okay, and that one's beautiful because, you know, if you put 3 proteins in a test tube and with ATP, it will be a circadian clock, basically. It will have a cycle of 24 hours, basically, which doesn't rely on the nucleus at all, okay? And we'll say then that erythrocytes must have something similar, okay? So our understanding of this is not complete. Right, then we'll end the video by discussing some examples of chronopharmacology. Okay, so we'll talk about how aspirin is best absorbed if you take it in the evening. That's an example of chronopharmacokinetics. And then we'll talk about an example of chronopharmacodynamics, which is that statins are most efficacious if you take them in the evening. Okay, and then we'll talk potentially about future instances of chronopharmacology. So, for instance, anti-cancer chemotherapy is where everyone's looking to. Okay, right. So let's start off then with what a circadian clock actually is. So we need to start off with what a circadian rhythm actually is. That's the basis for all of this. What is a circadian rhythm? Okay, right. So a circadian rhythm is some sort of cyclic process, okay, that repeats every 24 hours. Okay, so the most common sense example of a circadian rhythm is the light-dark cycles. Okay, so it gets light, it then gets dark again, it then gets light, it gets dark again. It goes on and on, basically. That is the most common sense example that I can give you of a circadian rhythm. Okay, it's a process which is going around on a cycle with a period of 24 hours. Okay, now, before we just move on to the relevance of this to biology, let me just tell you what circadian actually means. So circadian is derived from some Latin words, okay? The 2 Latin words circa and then diem. Okay, so what do these 2 mean? Well, circa is Latin for around, okay, and diem is Latin for a day. Okay, so circadian just means around a day, so it refers to the fact that these circadian rhythms are rhythms with periods that are around a day, basically. So that's all that this fancy word circadian actually means. It's just referring to the fact that these are 24-hour rhythms. Okay, right. So So we've seen this basic common sense example of a circadian rhythm, which is the light-dark cycle, okay? However, humans have circadian rhythms, okay? We all know this. Our behavior changes on a circadian basis, okay? We have the sleep-wake cycle. We are awake in the day, we then go to sleep. We are then awake, we go to sleep. We are awake, we go to sleep. This repeats over and over again on a 24-hour cycle, okay? So our behavior changes on a 24-hour basis. We are circadian, basically. Our behavior is a circadian rhythm. Okay, so humans, we know, have circadian rhythms, okay? So do loads of other organisms. What it might come to what might surprise you to find out is that actually it's not just the entire human which has circadian rhythms. In actual fact, all of your individual cells have circadian rhythms, okay? All of your cells within your body are changing their behavior on a 24-hour cycle, basically, okay? And that might come to surprise you, okay? And this isn't just humans, this is loads of different organisms, even single-celled organisms. Their behavior changes on a 24-hour basis. And you might say, well, surely that's just because they're responding to light-dark cycles. Okay, surely they are just responding to it being light and then changing their behavior because it has become light. Well, you can easily disprove that point of view by taking the organism, that maybe the single-celled organism, when we will see a single-celled organism later on, we're going to look at the cyanobacterial circadian clock in Seneca-Coccus elongatus. Okay, that's a single-celled organism. Okay, it hugely changes its behavior on a 24-hour basis. Okay, and you might say that's just it responding to light and dark. Okay, however, you can put it into an environment where it's totally dark, okay, all the time, and it continues to alter its behavior on a 24-hour basis, basically. It has a way of knowing what time of day it is and changing its behavior according to that, which doesn't rely on it being exposed to the light-dark cycle, basically. It's not a feedback system of the organism responding to the light-dark cycles. These behaviors are driven by a feed-forward system where the organism changes its behavior in anticipation of the light-dark cycle. Okay, so it's cleverer, basically. Okay, right, so that's the sort of general discussion of circadian clocks then, and in fact I should probably just define then what a circadian clock actually is. So basically, I've now let the cat out of the bag, the great sort of main theme of this video, which is that cells in the human body and cells in single-celled organisms and cells in all sorts of different organisms all have these oscillators, basically, these clocks that allow them to tell what time of day it is, some sort of biochemical process that is oscillating on a 24-hour basis, which then allows them to understand what time of day it is and respond according to what time of day it is. Okay, and these oscillating biochemical processes, these are what are meant when we say circadian clocks. Okay, so a circadian clock within a cell is some sort of oscillating biochemical process that oscillates with a rhythm of 24 hours and allows the organism to predict the light-dark cycle. Okay, and some people will call these circadian oscillators as well. Circadian oscillator, circadian clock, they're the exact same thing. Okay, in addition, you might also hear people call it the internal clock. Okay, that means the same thing as well. Okay, right, so now I think the best way to actually illustrate what a circadian clock actually is just launch straight into an example, okay? Because by seeing an example, we will begin to understand what I mean by a biochemical oscillator that allows you to tell what time of day it is. Okay, so the first circadian clock then that we are going to study is from Drosophila Melanogaster. Okay, which is a species of fruit fly. Okay, Melanogaster rather than Melanogaster. Melanogaster. Okay, Drosophila Melanogaster. Okay, right. And for short, you might sometimes hear or see Drosophila Melanogaster just abbreviated down to D Melanogaster. Okay, right. Okay, so Drosophila Melanogaster, then, it has a circadian oscillator inside all of its cells, basically. Okay, all of its cells know what time of day it is. Okay, and what's incredible about all of the cells of the Drosophila Melanogaster are that they are capable of synchronizing this clock with the light-dark cycle. Okay, so there is this terrible problem with circadian clocks, which is that how do you keep them synchronized with the light-dark cycle? Okay, because you don't want your internal clock launching a response that is appropriate for the daytime when it's light in the middle of the night. Okay, there is this problem that the circadian clock could end up out of synchrony with the light-dark cycle, and then the cell would be having the completely wrong behavior for the wrong time of day, basically. It might be getting ready for photosynthesis in the middle of the night, basically. Okay, and that's not what you want. You want it to get ready for photosynthesis at dawn, okay, when it's about to become light. Okay, so you need ways of keeping circadian clocks in synchrony with the light-dark cycle. And what's incredible about Drosophila Melanogaster cells is that all of them have the capacity to synchronize their circadian clock with the light-dark cycle. They can respond to light and synchronize their circadian clock with the light-dark cycle, basically, which is something that human cells are certainly not capable of doing. Okay, they rely on the master circadian clock, which is the suprachiasmatic nuclei, to synchronize their circadian clocks with the light-dark cycle. Okay, so without further ado then, let's actually see what this circadian oscillator or circadian clock is within Drosophila Melanogaster. Okay, so what is this biochemical oscillation which oscillates with 24-hour period? Okay, right. So to talk about this then, I need to introduce you to two important proteins within Drosophila Melanogaster cells. Okay, so the first important protein that I need to talk about is a protein which throughout this video will be abbreviating down to PER, okay, and its full name is period, however. So this is the period protein, and I'll highlight the period protein up here in green, okay. And then another important protein that has pretty much the same functional, analogous function to period, okay, or rather it works with period, is a protein called timeless. Okay, and for short we will be abbreviating timeless down to TIM. Okay, so these are the two stars, basically, of the Drosophila Melanogaster circadian oscillator, period and timeless. Okay, so these are proteins that are expressed within the Drosophila Melanogaster cells, okay, and their levels within Drosophila Melanogaster cells are going to oscillate on a 24-hour basis, basically. Okay, that is what the circadian oscillator is. Period and timeless levels go down during dawn and up during dusk. Okay, so let me draw you then a graph to show how period and timeless levels are going to oscillate throughout the day. Okay, so we'll be drawing lots of graphs like this throughout this video. So on the x-axis I'm going to be having time, and we'll be going up in sets of 4 hours, basically. So this is midnight, basically, 0 a.m. in the morning, 4 a.m., 8 a.m., 12 o'clock midday, okay, then 4 o'clock in the afternoon, 8 o'clock in the evening, and then we'll have back to midnight over here. Okay, and on the y-axis, what I am then going to put is the level of these two proteins, period and timeless. Okay, and their levels are going to oscillate together. Okay, so basically what I mean by that is the graph is going to be the same whether I'm drawing it for period levels or timeless levels. So I'm going to draw just one graph for both of them. Okay, right. So, we will begin at 8 a.m., okay, because that's about the time when period and timeless are lowest in the Drosophila Melanogaster cells. Okay, so we'll start off here with them being very low, and then throughout the day, their levels are going to be going up and up and up, okay, until around 8 o'clock at night, so 12 hours on, when their levels get to around the peak, okay. So really, in early morning, okay, you have the minimum level of period and timeless, and then right at dusk, when it's about to go dark at 8 o'clock in the evening, okay, then you have the maximum level, okay. Then what's going to happen is it's going to start to fall, okay, and it will be falling throughout the night. So here we've gone down to midnight now, and the period and timeless levels are going down. Now we'll continue the graph on this side here, okay, and it will fall down and down to get to that minimum then at 8 o'clock in the morning. Okay, so let me just colour this graph in to make it look smoother now. Okay, so that is the circadian oscillator inside Drosophila Melanogaster, or at least part of it, one of the crucial parts of it, which is that these period and timeless proteins are going up and down and up and down on a 24-hour rhythm, basically, inside all of the cells of Drosophila Melanogaster, this species of fly. Okay, right, so that's lovely. Okay, that's the way that we can tell what time of day it is if you're one of these Drosophila Melanogaster cells. Okay, what we now want to have a look at is what is it that actually produces this? Okay, why does this occur? And it's incredibly simple why it occurs. Okay, so in order to understand why period and timeless levels oscillate like this on a 24-hour basis, we need to discuss two more proteins, okay, two more stars, okay, and these are also extremely important components of the Drosophila Melanogaster circadian clock system. Period and timeless are one half of it, these next two are the other half of it. Okay, so the next two are two transcription factors. One is called clock, which we'll be abbreviating down to CLK, okay, and we'll be colouring clock in green throughout this video, okay, and then the other is called timeless, okay, and we'll be abbreviating timeless. Oh, sorry, we've done timeless. No, the other one is not called timeless, get rid of that. The other is called cycle. Okay, so there are these two transcription factors, clock and cycle, okay, and we'll be abbreviating cycle down to CYC. Okay, right, so these are these two transcription factors which are going to be extremely important in controlling this Drosophila Melanogaster circadian oscillator, basically. Okay, right, so clock and cycle then, what do they actually do? So they're transcription factors, and they work together, basically. They aren't transcription factors on their own. They have to form a heterodimer with one another. Okay, so they're going to form a clock cycle heterodimer, which I'll be representing like this. Okay, so we'll have clock represented here in green. Okay, and then we'll have cycle represented here in blue. Okay, so these two squares, these are the clock and cycle proteins heterodimerized together, so I'll just label them up. So this is clock here, okay, and then we've got cycle sitting next to it. So we can call this thing a clock cycle heterodimer here. Okay, so clock and cycle then, they are these two proteins that form this heterodimer, and this heterodimer is now going to act as a transcription factor. So let me just tell you about what transcription factors do. Okay, so transcription factors all have special sequences of organic bases which are generally between 4 and 10 base pairs long, which they can recognize in DNA and bind to. Okay, now the special sequence of organic bases in DNA which clock cycle heterodimers can bind to and recognize is a sequence called the E-box. Okay, now I do not actually, unfortunately, know the exact sequence of organic bases that the E-box actually consists of. Okay, but it will be a sequence of organic bases between 4 and 10 base pairs, which is specific for this clock cycle heterodimer. Okay, so this is called the E-box here. Okay, so there is this special sequence of organic bases then that clock cycle heterodimers are capable of binding to. Okay, now, That's lovely, that's what transcription factors do. They bind to DNA, but what does that actually do? Okay, well, transcription factors, you probably know, are incredibly important in controlling the transcription of genes. Okay, so let me draw another picture now, here. Okay, so once again, this represents a double-stranded piece of DNA here. Okay, and let's say that this portion that I'm colouring in orange here, this is some gene. Okay, so this is a gene. Okay, now upstream of all genes in the eukaryotic genome, which of course Drosophila Melanogaster cells are, they are eukaryotes. Okay, you have a region called the gene control region. Okay, so this portion here is going to be called the gene control region. Okay, and the gene control region controls how often the downstream gene is actually going to be transcribed. Okay, and by controlling how often the downstream gene is actually going to be transcribed, we can control the actual amount of protein from that gene that you produce, basically. Okay, because by controlling how much transcription occurs, we control how much mRNA for that gene you produce. By controlling how much mRNA you produce, we control how much protein you produce. Okay, so quite simply, control of transcription controls the protein expression of the gene. Okay, right. So, basically, the special sequence for a transcription factor, such as the E box in the case of the clock cycle heterodimers, will be found in the gene control region of a bunch of genes. Okay, so for instance, the E box will be found in the gene control region of maybe up to 100 or beyond genes in the Drosophila genome, basically. Okay, and when clock cycle heterodimers bind to that E box, they can affect the transcription of the downstream gene. Okay, and clock cycle heterodimers tend to increase the transcription of the downstream gene. Okay, so here we might have an E box in the gene control region of this gene. Okay, and what then will happen is a clock cycle heterodimer can bind to that E box. Okay, and it will then result in the promotion of the transcription of the downstream gene. Okay, so it's going to increase the amount of transcription that occurs for the downstream gene. Okay, so that means now that more mRNA will be produced for that downstream gene, and therefore the expression of that downstream gene is going to go up. So basically, clock cycle heterodimers can drive the expression of a bunch of genes. Okay, now two of the genes that are relevant to us that these clock cycle heterodimers increase the expression of are period and timeless. So both period and timeless genes both have E boxes in their gene control regions. Okay, and these E boxes are going to bind clock cycle heterodimers. Okay, and when the clock cycle heterodimer binds to that E box, it will increase the expression of the period and timeless genes. Okay, right. So now we've got all the sort of prerequisite stuff. Let's now put it all together into a great big system. Okay, so we're going to start somewhere with this cycle, and we're going to start at 8 o'clock in the morning, when period and timeless levels are really low within the cell, basically. Okay, so period and timeless are very low at 8 o'clock in the morning, and that's where we're going to start. Okay, so when period and timeless are low, it turns out that clock and cycle are very active at that time. Okay, and you'll see why later on. Period and timeless are going to turn out to inhibit clock and cycle heterodimers. Okay, so if we've got the low period and timeless, then the clock cycle heterodimers are going to be very active, basically. Okay, right, so I'm wondering now whether I'm going to try and squeeze my picture in here or go over onto the other page. I think I'll go over onto the other page to show this next picture, because it's going to be a big picture. Okay, so I'm now going to draw the nucleus of my Drosophila Melanogaster cell here. Okay, right, and what I also want to put here is the genes for period and timeless. Okay, so this is, if you like, the main picture that's going to summarize the circadian clock system in Drosophila Melanogaster cells. Okay, right. So here we'll have the gene for period shown here. Okay, and here we'll have the gene for timeless shown here. So I'll just label them up. So this is the period gene, okay, represented by those two parallel lines coloured in orange there. And I've left the gene control region deliberately in on this picture. Okay, and here, this down here, this is the timeless gene. Okay, right. So, at 8 o'clock in the morning then, period and timeless levels within the cell are very low. Okay, and I've said, for reasons that we will see later on, that means that the clock cycle heterodimers are all free in the nucleus and are active, basically. They can bind to E-boxes and increase the expression of the downstream genes. Okay, I've also told you that both the period and timeless genes have E-boxes in their gene control regions. Okay, so what this is now going to mean is that clock cycle heterodimers are now going to bind onto our E-boxes here. Okay, like so. Okay, so let me colour these in. Okay, so here are the clock proteins in green there. Okay, and here are the cycle proteins in blue here. So the clock cycle heterodimers bind to those E-boxes upstream of the period and timeless genes, and they are now going to increase the transcription of those genes. Okay, so this is occurring at 8 AM, so in the morning basically, you're now going to be getting lots of mRNA produced for the period and timeless genes. Okay, so let me now represent this here. So the mRNA will obviously leave the nucleus to be translated, and it will go into the cytoplasm. Okay, so here it is going into the cytoplasm out here. Okay, this in orange here, this is the period mRNA. Okay, and this in turquoise here, this is the timeless mRNA. Okay, now what will obviously happen is the period and timeless mRNAs will both be translated into period and timeless proteins. So now, period and timeless proteins are going to start going up in the cytoplasm, and that's exactly what we wanted to happen. Okay, you'll remember that on that graph, period and timeless go up during the day. Okay, so we started off at 8 AM. Okay, with low period and timeless, that has resulted in clock cycle heterodimers being active, okay, and they are now driving an increase in the level of period and timeless proteins in the cytoplasm. Okay, and I'll colour in the period and timeless proteins here once again in orange and turquoise. So here is my period protein here in orange, and here is my timeless protein in turquoise. Okay, right. So this is going to be happening throughout the day, basically. Okay, clock cycle heterodimers from 8 AM onwards are going to be driving the increased expression of the period and timeless genes. Okay, and this is why the levels of period and timeless are going to go up during the day. Okay, until 8 o'clock in the evening, when they reach a maximum and then start coming down. So why does it start then turning? Why do period and timeless levels not just keep going on, up and up and up? Okay, well, basically what starts to happen is period and timeless proteins start to form heterodimers. Okay, like so. So they start to dimerize together like so. So you get one period protein and one timeless protein coming together to form a period timeless heterodimer here. So here's the timeless protein in turquoise, here's the period protein in orange. Okay, and this takes a long time. I want to stress this. This is a delayed bit of this pathway, so I'll colour these in blue. This is delayed, okay? It takes a long time for period and timeless proteins to actually start to heterodimerize, and I'll talk a little bit more about why that is in a moment. Then what can start to happen is period and timeless heterodimers can go into the nucleus, and again this is a really delayed process. Okay, so you might be making these period and timeless proteins at midday in the day, okay, but they're not actually going to start arriving in the nucleus as heterodimers until 8 o'clock at night. Okay, so this is the point. Okay, 8 o'clock in the evening, I should probably say, rather than in the night. This is the point where period timeless heterodimers actually start entering the nucleus. Enter nucleus. Okay, and when they enter the nucleus, they're now going to stop their own transcription, basically. Okay, and we will discuss why it takes them so long to actually get into the nucleus in just a moment. Okay, so you've been making them from 8 AM in the morning. Okay, why has it taken them so long to actually heterodimerize and get into the nucleus? Okay, right. I will discuss that now, actually. I'll come back to them, what they're going to do in the nucleus in a moment. So why does it take so long? Why is there this great delay? Okay, so we now want to ask about this delay. Well, the answer is we're not absolutely sure, but we do know something. Okay, so it's not the case that we know nothing, but we don't know it fully. Okay, so basically it seems that in order for the period and timeless proteins to actually heterodimerize, you have to make loads of modifications to them, basically. Okay, you have to make loads of post-translational modifications to the period and timeless proteins to get them to heterodimerize. Okay, it seems also that you have to make lots of post-translational modifications to the period timeless heterodimer then to actually get it into the nucleus. So basically, the answer is that my picture is too simple. Okay, it's not the case that you just make period and timeless proteins, they bind to each other and then they go into the nucleus. There are loads of little tweakings, loads of faffing that is occurring, loads of little modifications that are being made to the period and timeless proteins, both before they can actually even heterodimerize, and then also to the heterodimer in order to allow it to get into the nucleus. So that is why it takes so long. That's why it takes 12 hours, basically, to get the period and timeless proteins actually ready so that they can go into the nucleus. Okay, so we know about certain enzymes then in Drosophila that seem to be extremely important in making these post-translational modifications which are needed in order for period and timeless heterodimers to actually heterodimerize together and also then end up in the nucleus. Okay, so some important ones then. What we have seen big time is that phosphorylation seems to be a major post-translational modification. So phosphorylation is one of the major post-translational modifications that seems to need to be made. Okay, and we have found enzymes that are really important in phosphorylating period and timeless proteins. Okay, so that they can heterodimerize and then so that they can enter the nucleus. Okay, so if you disturb these enzymes, then things go horribly wrong with this process, basically. Okay, so we know that they must be involved either in allowing the period and timeless to heterodimerize or in their translocation to the nucleus. Okay, so some important enzymes then that are involved in these post-translational modifications which are causing this delay here, okay, are, for instance, an enzyme called double time, okay, which for short is abbreviated down to DBT. Okay, so this stands for double time. Okay, and this enzyme is involved in phosphorylating the period protein. Okay, so this is a period protein phosphorylating enzyme, or a kinase enzyme for the period protein. Okay, there is another enzyme which is important in phosphorylating the period enzyme, okay, which is called casein kinase 2. Okay, and for short I'll abbreviate casein kinase 2 down to CSK2. And I'm sorry if this is seeming awfully listy, okay, it's because we don't fully understand this yet. Okay, we know that there's a post-translational modification causing this hold-up here, okay, but we don't know the exact orchestration of that yet. All we know is some enzymes which are important in catalyzing those post-translational modifications, and that's what I'm giving you at the moment. Okay, so double time is really important in allowing period to actually either heterodimerize or for it to go into the nucleus. Okay, and so is casein kinase 2. Okay, CSK2. And we'll see in the mammal actually where there's going to be a very similar circadian clock system to this, that casein kinase enzymes are important there as well. So casein kinase 2 in Drosophila Melanogaster cells is important in phosphorylating period as well. So both double time and casein kinase 2 phosphorylate period. Then there is another enzyme which is important in this faffing about here, okay, which actually does the reverse. It removes phosphate groups, okay, and this is protein phosphatase 2A, PP2A, okay, so this stands for protein, that's the first P, and then phosphatase, that's the second P, and then specifically protein phosphatase 2A. So it might be the case that these ones put it on, this one takes it off, and then it's a faffing around, waiting for one of them to get the better of the other. We don't know, basically, but something is causing a great big amount of faff here to actually get the period and timeless proteins to heterodimerize and to translocate into the nucleus. Okay, so all of those are about period. Okay, these phosphorylate period, this one dephosphorylates period. Now let me give you one enzyme that's involved in timeless, and I'll put this on the top here. Okay, so there is an enzyme called Shaggy, which is basically known to phosphorylate timeless. Okay, so that is a great long list of enzymes then that are involved in this post-translational modification phase that is needed to make the period and timeless proteins heterodimerize and translocate into the nucleus, basically. But at the moment we don't know the exact orchestration of this. We don't know what is really happening there. This is just some enzymes that we know are involved in that process. So I'm sorry if that seems awfully listy. It's because it is, and that's the best I can do, basically. Okay, right. So that's what causes this great big delay in the period timeless heterodimers actually getting into the nucleus. Okay, so at around 8 o'clock at night then, they will actually now be starting to arrive in the nucleus. Okay, now what do they do once they're in the nucleus? So let's just draw it now in the nucleus. Okay, well, the basic answer is they inhibit the clock cycle heterodimers, basically, okay, and stop them from activating the expression of all the genes with E boxes within them. Okay, so here is our period timeless heterodimer here, and it now is going to stop the clock cycle heterodimers from activating the transcription of genes with E boxes in their gene control regions. Okay, now there is still a bit of controversy here as to whether it is the period timeless heterodimer which is responsible for inhibiting the clock cycle heterodimer, or whether it's just the period. Okay, some people believe that what happens is when the period timeless heterodimer gets into the nucleus, it then breaks apart again, and then it's period alone which then inhibits the clock cycle heterodimers. Okay, other people believe that it's the period timeless heterodimer that functions as one and inhibits the clock cycle heterodimer. I'm going to draw it as though it's the period timeless heterodimer that inhibits the clock cycle heterodimmer, simply because it looks more even. Okay, but it may turn out that it's the other way around. Okay, this is still controversial. Okay, so what now happens is when these period timeless heterodimers arrive in, what they're going to do is bind to and inhibit the clock cycle heterodimers. So here now is my clock cycle heterodimer, sequestered away by these period timeless heterodimers. Okay, so going back to this graph then, at 8 o'clock at night, this is what's going to be happening. Period timeless heterodimers have got into the nucleus, okay, and they are now sequestering and therefore inhibiting the clock cycle heterodimers. So clock and cycle are no longer functional. Okay, right, so what's that going to mean? Well, that means that we are now going to get no more production of period and timeless proteins. Okay, period and timeless genes have now been turned off effectively, because remember what was turning them on was the clock cycle heterodimers. So period and timeless inhibit their own expression basically through this mechanism that we have seen here. Okay, so no more production of period and timeless is going to occur. Now what's going to occur is in the cytoplasm, period and timeless proteins that have been produced here are going to start being degraded, okay? And that is why you start getting this downfall, basically, okay? So now that you're no longer adding to the store of period and timeless proteins, degradation is going to take over, okay, and you're going to see net degradation, okay. So previously there was degradation obviously active, okay, period and timeless proteins were being degraded, but because we were producing new ones and we're producing them at a rate larger than the rate at which they were being degraded, then period and timeless levels were going up in the cytoplasm of the cell. Now, we've stopped producing any new ones, so the degradation is going to take over, and therefore period and timeless levels are going to go down in the cytoplasm. So we're going to lose all of the period and timeless in the cytoplasm that we have created, okay? And that's what's occurring in this down phase here, from 8 o'clock in the evening to 8 o'clock in the morning. Period and timeless proteins are being degraded in the cytoplasm. Now again, there is some great delay here, okay, because even though period and timeless levels are going down in the cytoplasm, it takes an incredibly long time for that reduction in the cytoplasm to then actually translate to a reduction in the nucleus. So the period timeless heterodimers that have actually made it into the nucleus, it takes an incredibly long time for them to actually be degraded, basically. Okay, so the ones in the cytoplasm fall, but there is then some great delay about getting rid of the ones in the nucleus. Okay, so that they only really start to be removed towards the bottom here. Okay, so towards 8 o'clock in the morning now, the period timeless heterodimers in the nucleus are being removed. Okay, and that now means that the clock and cycle heterodimers will be released at around 8 o'clock. So at 8 o'clock then, period and timeless will have gone from the nucleus, okay, with that great delay, okay. So period and timeless were being degraded in the cytoplasm all through the night, but then there was some great delay about getting the period and timeless out of the nucleus, okay. By 8 o'clock in the morning we have done it, okay, and therefore our clock cycle heterodimers are now once more free in the nucleus, okay, and then you can just begin it again. We're right back where we started. No period timeless, clock and cycle are all free, and then we can just go around this exact same cycle again, basically. Okay, and that goes round and round and round, and that is the circadian oscillator then that is present within the Drosophila Melanogaster cells. Okay, right, so just some other things to say. How does this actually then influence the cell's behaviour? Brilliant. This is a beautiful biochemical oscillator. It's a circadian clock that is present within the Drosophila Melanogaster cells. But how does this actually change their behaviour? Well, basically, the thing is that these clock cycle heterodimers, okay, these really important transcription factors, they will be transcription factors for other genes as well. So let me add in another gene here. Okay, so this final gene here, which we'll have in vivid purple, this can be some effector gene, basically. Okay, some effector gene that you want to be expressed during the day, basically. Okay, and upstream of that effector gene, we can have our E-box here. Okay, and then from 8 a.m. all through the day till 8 p.m., you're going to have clock cycle heterodimers being free in the nucleus. Okay, and these clock cycle heterodimers will be binding to that E-box and promoting the expression of that effector gene, so that the effector gene's level is going to be high during the day, and then at the night, because the period and timeless heterodimers have entered the nucleus, you're going to be getting the inhibition of clock cycle heterodimers, and therefore the expression level of that effector gene will drop. Okay, so if that effector gene is a gene that we want expressed in the day, then we can now see how these circadian clocks are actually going to determine what the cell is doing, basically. Okay, so by controlling the gene expression of a huge number of effector genes, we can change the behavior of the cell so that it has one behavior during the day and then a different behavior at the night. Okay, so that's how this circadian clock system can actually change the behavior of the Drosophila Melanogaster cells. Okay, right. So what I now want to talk about is, finally, how can they make sure that this circadian clock system is synchronized with the light-dark cycles? Because nothing about what I've told you yet means that this system should remain perfectly in synchrony, so that you've got the low period timeless at 8 a.m. and the high period timeless at 8 p.m. Okay, at the moment you could shift this and it would still work perfectly. There's nothing about light and dark here at all. Okay, so why does it stay like this? Why doesn't this end up shifted somewhere? Okay, why are all of the Drosophila Melanogaster cells fluctuating with it like this, where period and timeless are lowest at 8 a.m. and highest at 8 p.m.? Okay, well the answer is that they have a mechanism to synchronize the circadian clock to the light-dark cycles. Okay, and this involves one final protein that I'll introduce you to now. Okay, so this involves a protein called cryptochrome, which we'll be abbreviating down to cry. Okay, so cryptochrome. Okay, and basically cryptochrome is a protein which can respond to light. Okay, so light is going to influence the behavior of cryptochrome. So during the daytime, light is going to be activating the cryptochrome protein. Okay, and the cryptochrome protein is then capable of leading to the degradation of period and timeless proteins. Okay, so if I add this onto this picture here, here is cryptochrome, and it's capable of accelerating the degradation, and I'll put inhibiting basically here. Okay, it's capable of inhibiting the period and timeless proteins. It's capable of making them be destroyed in the cell faster. Okay, now let's think about what that is actually going to do then. Okay, so let's think about what would happen if during the early evenings, let's say during around 7 o'clock maybe, when we're going towards dusk, what would then be the effect of exposing a Drosophila Melanogaster cell to bright light? Okay, well what would of course happen is we'd activate our cryptochrome protein. Okay, it would now accelerate the degradation of period and timeless. So what would happen to our period and timeless levels? They would be moved down, maybe to here. Okay, so effectively it would move the clock back towards the middle of the day, basically. Okay, you'd be moving the period and timeless levels down, basically, and now the clock would be reset to somewhere maybe like here. Okay, so maybe 2 o'clock in the afternoon, so you'd be moving the clock back, and that's exactly what you want to do, because you've just received bright light. That's telling us that the clock is wrongly set, basically. It surely is the middle of the day rather than 7 o'clock at night. Okay, so if the circadian clock thinks that it is 7 o'clock at night, and in fact you're receiving bright light telling you that it's the middle of the day, it will reset the clock and move it backwards, okay, so that the clock's time is now set to a more appropriate time in tune with the light-dark cycle, basically. Okay, so if the circadian clock thinks that it is early evening, okay, so maybe the circadian clock thinks that it is 7 o'clock in the night, but then you receive bright light which indicates that the light-dark cycle might be closer to midday, then the clock will go back, okay? The circadian clock will slow down and it will move back, basically. Okay, so that's what cryptochrome will do to the circadian clock in the early evening, or at least if the circadian clock thinks that it's early evening. Okay, obviously if we're receiving bright light, then it's the middle of the day most likely. Okay, let's think about what will happen if we do it in early morning rather than early evening, so maybe 4 a.m. in the morning or something like that. Okay, so if we now go to 4 a.m. in the morning, or maybe let's make it earlier, 2 a.m. in the morning, okay, and suddenly you get bright light, what will that do? Well, the circadian clock thinks that it is 2 a.m. in the morning, okay, however, if you've received bright light, that tells us that the circadian clock is wrong, basically, it's out of tune with the light-dark cycle, okay, so we need to move the circadian clock forwards, basically, towards daytime, okay. So what will happen is the cryptochrome proteins will be activated by the bright light, okay, and they will then accelerate the degradation of period and timeless proteins, and that will now move the clock forward. So if you can imagine, it will go down, and that's going to move us forward basically to a later time, a later circadian time, a later time for the circadian clock. Okay, so you'll move the circadian clock's time towards thinking it's more daytime, basically. Okay, and this then is how you can keep your circadian clock in time with the light-dark clock, basically. Okay, right. So early morning, we're going to set the clock forwards, basically, if you suddenly receive bright light, to move it so that the circadian clock thinks that it's more middle of the day. Okay, so bright light suggests that it's the middle of the day. If we are thinking it's early night, then we're going to be moving the clock backwards. If we think it's early morning, okay, we're going to be moving the clock forwards. Okay, so that's how we can keep our circadian clock system in tune with the real clock system, which is the light-dark cycle. Okay, right. So we'll call it there for this video, and in the next video what we'll move on to is the mammalian circadian clock system.